So this is the end. So we started back in February now, which seems like a long time ago, with the absolute basics, and then we've gone through all the stages of developing an LLM project. So from getting your data to the choice of framework, and then some example projects. So all the way through, we've had really great questions, and I've really enjoyed answering them. But as is always the case, we always run out of time to answer everything. So what we thought we would do is have this whole session just purely dedicated to answering questions that came up throughout the webinar series. So if you've got a question about anything for any of the panelists, we're very lucky to have everyone here today. I'm really pleased and really thank you all very much for coming. If you've got a question for any of us, please pop it in the Q&A. If you see a question you like the look of, upvote it, as AJ says. One thing I will say is we've only got Sebastian for the first hour. So if you have BioChatter specific questions, or you just want to pick Sebastian's brain on something, get those in first, because he's got to go halfway through. But the rest of us will be around. So I see we've got one already. We'll ruminate on that for a second while I ask one of the questions that we had from the pre-submitted few that we got. Thank you for those. So this is quite an interesting one, which I've been thinking about lately. So what do we think for new AI systems, what do we think the peer review process should prioritize? Should we be focusing on evaluating the conceptual framework or the application or the underlying code? Or should it all be a kind of holistic interpretation? And how would you review in the most effective way the current state of LLMs and things? Does anybody want to chime in? Okay, go on, Sebastian.
I think this is on many people's minds because the staggering pace that we see, particularly driven out of industry with very specific applications, and the companies that basically try to get into every nook and cranny of society with these technologies. This leads to not being able to follow anymore, which is maybe an indirect answer to the question, but the main motivation for this a priori benchmarking philosophy that we have, which is we need to set up ideally with domain experts. So, for example, with doctors when it comes to medicine, or with structural biologists if it's about protein folding or something like this, set up somewhat of a Turing test, but not as simple as the Turing test for the domain that we're interested in, and then find a way to dynamically probe the models in a continuous integration way. Because tons of models coming out all the time, we cannot deal with them in the manual, I'm going to have a look at this and then I'm going to try this kind of way. I think we need to define what we want them to do a priori and then find smart ways to inflict this upon the LLMs. And then it doesn't matter if 200 LLMs are released in a given month. As long as you have the compute to evaluate, I think that is a more sustainable way of approaching it.
So would you say if you were given a paper that's developed an LLM application to review, would you want to see its performance on some independent benchmarks as part of that paper, or would you
Well,
reject it if they
yeah.
didn't have that sort of thing in?
It depends. So if you say papers, I was referring to models right now because it's simpler in
Mhm.
that case. With papers, you can have a bunch of different avenues. Maybe they just develop a new type of model. Maybe they specify it for a certain domain, and this alone would be two very different use cases. For the domain, I would expect them to have domain experts involved and define success metrics that are closely aligned with the goals of this domain. If it's a generic model paper about some interesting architecture or a mechanism in them, then it can be maybe more in the sense of a computer science conference where you just have other experts in computer science look at it. So I guess that's the scope of papers is much bigger than the scope of models.
Yeah. Okay. Melanie?
Yeah, I think I broadly agree with Sebastian. I think what we need is a sort of an automated framework with benchmark data sets for specific domains and just then automatically test models against it. If you have a paper and so I've had occasionally not LLMs but other machine learning algorithms, I had papers to review and I generally I look at the statistics, the performance, I make sure that the data is public so that I can actually check whether it's sort of personalized data or whether it's open accessible, whether I can have a look into the data if I want to. But for example, I would never review the code base because that's just far too large. In most cases, the code around it, the proper GitHub repository is far too large. On the other hand, if they provide things like an API for a model, or yeah, or some other way of, having a quick way of installing it, downloading it, and with some test data and running it. I do that and see if I can reproduce some of the results that they publish, if the data is available as well. But I'm not going through every single line of code. I wouldn't do that. And yeah, as Sebastian said, I would always expect that for certain applications, the computer scientist will link with the biological scientist for a topic, and not just develop an algorithms because they can do it. Yeah, I expect it to be teamwork.
Yeah, yeah. So would you, for example, if someone's developed something around a closed model, like they've just used OpenAI's API, for example, how would you, what would you want to see from like as a reviewer for a paper that's heavily built on something that you can't just, where you can go against it, but you need to spend big money potentially to do a reproduction?
Um, it's a good question. I haven't been in that situation yet. So what would I do? To be honest, I would probably ask to find a different reviewer because I do not want to support OpenAI. I refuse to get
Mm.
myself a paid account. So if I can do something that's within, say, ChatGPT, I will try. Again, test examples they provide. If there is no test example, if there is no alternative way of doing it, I probably would refuse to review the paper because I don't think it's appropriate for open research. I've had other people sending me emails going, oh, we've done this fancy model. Do you want to give us a go? It's for PDB data. And as soon as I see an email, I need an OpenAI key or I need to pay in some way. I'm not using it.
Hmm.
Sorry, that's my personal view.
Yeah. So I saw Matthias, you had your hand up. Did you have something to add on that?
Well, it was before the talk about OpenAI and refusing to use OpenAI, or yeah, how to address the or to review a paper that use a closed source model. It's more about the benchmarks. It's when reviewing a paper, if the authors submit an evaluation of the model, you have to also take a bit of pinch of salt because they can tweak benchmarks. For instance, last week there was Llama 4 that was released. Everyone had a huge expectation on the performance of Llama 4, and now Meta is accused, let's say, to have kind of tweaked or boost benchmark AI benchmarks to have Llama 4 ranked higher. So I think what it's better. It's difficult to create, but it's better. It's maybe a community-driven platform to evaluate large language models to which authors could submit their models. They need to be evaluated there, maybe privately at first, so the model is not released until the paper is published. But at least it gives a way for reviewers to evaluate the model independently. That it was just these comments.
Hmm. Okay, thanks. Sebastian.
Yeah, that was kind of in response to both of these points. So I've been in the situation a couple of times where I got such a paper, and then it depends on the way that the authors have chosen to implement the performance testing. And the worst case, obviously, is they just built chatbot for something and they don't test it at all. So they create a web app and they say it should work, right? We tested it with 3 people in our lab and it went fine. And then obviously I don't refuse to review. I just say in my review, I will not buy this if you don't have some test battery that at least ensures basic functionality. And then it doesn't matter if it's OpenAI or other proprietary providers or it's an open source model. I don't need to host it myself. I need just to be convinced that the authors have some quality assurance in place that doesn't make the model run amok with the whatever questions you ask it, right? If you have a model that is supposed to support chemoinformatics, it shouldn't give answers to the best chocolate cake recipes. And these are some very basic things that everybody can do if they just go to the app and then check if the model behaves as it should. And for the benchmarking initiatives, it's kind of the same. So I really like this Apple paper on GSM symbolic, which is a symbolization of a GSM benchmark. So it's math reasoning problems, but the problems are not static, which facilitates hacking the benchmark and maybe overfitting your model developments to make it look good in the public benchmark. But rather, it allows you to create 1,000 different variants of the same problem with different numbers, different phrasings, maybe adding a sentence that doesn't really have any bearing on the problem and that wouldn't confuse a human student at all. But LLMs, for some reason, break down in their capacities. And that's what I meant by the a priori definition of the functionalities. You set out these things, like the LLM should be able to solve GSM level problems. And then if you have a symbolic benchmark that works, it should provide you with a more solid baseline than these memorizable public benchmarks that often only last one generation of LLMs.
Yeah, because like that's the danger with benchmarks and having them open is that they definitely leak into the training data and then they're useless. Yeah. Thank you all.
But also, I
Go on.
just had another insight ad hoc. In biology, it is really not straightforward to do it in the same way as in, for example, math reasoning, because we have a lot less ground truth. And so coming up with these benchmarks is really an art at the moment. And there are some initiatives, for example, at Helmholtz, to bring this more into the direction of a robust science. So benchmarking in itself is now becoming a scientific discipline with all the complexities that this entails. So it's not by any means a simple question to answer, but that also means that we have a lot to do, which always excites us as scientists.
Yeah, that's good. We won't be out of a job just yet. So this one I'm going to ask now because it's a BioChatter one for Sebastian. So for someone who would like to use BioChatter to build out an application for internal use in a bioinformatics core, could you explain the skill set needed to do this? I know building out applications uses a different tool set from bioinformatics pipeline development and analysis. Also, could you provide an estimate of the time that would be needed to invest to get up and running? And how is the continuing maintenance? How does that
Yeah,
look?
that's a good question and one I get quite often. And the best answer, as always, is it depends. If the application is very aligned with what we have already done, and this is one of the principles of the ecosystem, reusability, then you might be lucky and you just need to change a couple of parameters or the knowledge that is in the background, or maybe a bunch of text that instructs your user. And then there are things that are further out that we haven't attempted, maybe. And for that, you would need some skills in LLMs, obviously, and applying Python frameworks in general. And then it depends on what you want to provide your users with. So if you want to provide them with like a very fancy ChatGPT looking interface, then you probably also need someone that does something like web tech, JavaScript. If you are able to provide a simpler tool, then I would recommend going with these light implementations, which you only need to know Python for. And the same goes kind of for the duration of the project. A prototype should not take long if you're capable in the programming domain. And we also help with that. So if there is any projects that you would like to do, feel free to reach out. If you're able to share them, that's even better because then we can provide answers to the next generation of people that try to do it. And maybe your example is something that someone else also wants to do. And you can get as fancy as you want. So you can make this also a year-long project if it's about agent systems that are supposed to help with something very complicated. So there is no upper bound. At least I would plan for onboarding and everything 3 months for a simpler project.
Okay, thanks. That definitely sounds a lot quicker and easier than starting from scratch. Definitely. Okay, so you mentioned agents there. We've got one of our questions from the audience today is about agents. So in recent times, there's a lot of hype about LLMs as agents when we connect them up with bioinformatics tools. Um, how effective are the LLMs with and without tools at doing bioinformatics tasks? And how do we feel about agents in general?
So obviously we are very, very attentive about the agents because this seems to be the mode of choice that a large part of the industry has chosen to go about their solutions now. And there's also large players involved in defining these environments, technologies that make it easier, just to mention the model context protocol from Anthropic that was also adopted by OpenAI now, and probably most other large developers as well. And it is very attractive because it also allows you to synergize in an open source manner. So in our projects, we're thinking about building a model zoo. So model context protocols for everything we do in bioinformatics, for example, in the open target space, so that we don't need to define these tools at many different locations, but rather we have a centralized repository of the tools. The model context protocols are available, and then you can just kind of modularly put them together depending on which tools you want your models to be able to use. And if the question is how I feel about them, uh, I don't think they are there yet for bioinformatics because we make a lot of use of this intelligence in here to synergize with all the different steps that are involved in a typical bioinformatics question. And piecing together a bunch of agents that take care of all of these different things that we do is very difficult, particularly if you want them to select the right tool for the right job with an open-ended question. You have tons of tools, and each tool has tons of parameters, and this overwhelms the LLMs, at least in our experiments. So if we don't find smarter ways of organizing this, maybe in a semi-deterministic structure and very dedicated way, so what is the exact problem that we're trying to solve? Then I don't think we can just throw a bunch of MCP definitions at an LLM and expect it to work out of the box. So again, still a lot of work to be done.
Thanks, Melanie.
Yeah, I mean, again, Sebastian makes here the first comment, but it's, I think if you have a very well-defined problem where you can really, you have no lots of ifs and buts and no lots of alternatives that could be reasonably explored, I think you're going down with an agent route is probably a good way of sort of narrowing down what you have to do and really fix it onto, one implementation. But as Sebastian said, because it's not in biological field, it's not that you have just a single solution to a problem. There is lots of things where, if you just think if you try to build something that predicts your pathways in a cell, as soon as you change a parameter in a cell, say a pH or a temperature or something, your pathway will respond in a different way. And if you want to analyze something like that or model something like that using agents, you probably just kind of like it will just not be able to consider all of the different options that there are. And therefore, yeah, if you have something that's very nicely defined, you can, I think they're really helpful, but the complexity of the problem is just, yeah, overwhelming.
Yeah.
And if I may, also again, there's a lot of hype, which means the field is moving a lot. Which means if you pick a solution right now, where we're kind of in the early stage of AI models applied to fields such as bioinformatics, biology, or chemistry, there's a high chance in 6 months could be completely outdated and something else is going to come and replace them and do a much, much better job, which means you have to go back, maybe change your entire system, if not your stack, and again do all the testing, onboarding your users. So it's very appealing to just jump right now and start building tools. But yeah, as Sebastian mentioned, we're pretty not there yet. We need to wait a bit for big players to develop something more stable that can be then adopted by more domain specific fields such as bioinformatics.
Just to clarify, I wasn't really advocating for waiting. Um, you should spend your time with something that's useful, but I think it is really, it requires a lot of knowledge of the field to decide what is worth developing right now. And for example, figuring out model context protocols for very commonly used tools will not be too early because once the society has decided on a protocol to follow, the implementations can change if the protocol remains stable. And same goes for metadata annotations. If large players now decide that Croissant is the best metadata format to describe machine learning datasets, then it's probably fine to look at your datasets and describe them already in Croissant. And then maybe some changes come in a couple of months. And it will not be in vain to have described it already. But yeah, creating a full stack solution with something that is very hot at the moment, probably not a good idea. So uh, I fully agree with that.
Yeah. And so for my part, I've been building agents without agency. So agents that can just follow a predetermined plan. So it's not an agent really, it's just following the steps, which works really well because you don't let it wander off and do random things. And there's sometimes where it needs to choose a tool, but you can kind of whittle down the number of tools that are needed at every step. And it makes it much more tractable at the moment. If you want to build something now that will work, that's possibly a more tractable way to do it than just wait for the LLMs to get better. Um, I also worry that this sort of thing, I don't trust Anthropic and OpenAI and people to open source the underlying software that's going to make this possible. Because the potential for them to make an enormous boatload of money out of it is too great. And so people like DeepMind have been embargoing their research now because it's a competitive advantage for Google to not just publish everything. They published the attention is all you need paper, and then no one really predicted what was going to happen as a result of that whole thing. So they keep playing their cards a little close to their chest, and I worry that because open source has been catching up quite fast to the closed source things. I worry that maybe if we sort of just trust them to publish these things, that we might not get it, and someone will just have to build it open source. But then we're waiting even longer. So yeah, I don't really know where that leaves me, but I like building stuff and open sourcing it. So yeah,
Yeah, and this brings together these 2 points really. The first thing that you said about the more deterministic agents and the open source problem, because I'm not sure if you use constraint generation in these
yeah,
or if you because you can't do it with the model that you
no,
only connect to via an API, right? For constraint generation, you need access to the inner workings, and that is only possible if you have at least an open weight model that you can deploy yourself. And so I guess these things go kind of hand in hand. And organizations such as EMBL-EBI, Helmholtz, like the players that are big enough to at least be in the same sphere as large companies, I think they have some obligation to steer the discourse and the developments in this direction to be somewhat of a counterbalance to the very turbo capitalistic processes that we see going on in the large companies.
yeah, I'd agree with that 100%. Okay, I think we've covered everything on agents for now then. Let me just see. There's a question about public ontologies being used as a knowledge graph for LLMs. I mean, yes,
Yes, they can
yes,
be turned into knowledge graphs. They are basically knowledge graphs. You just have to, yeah, download them as they are and then turn them into a knowledge graph, and you can use them to support an LLM. Yeah.
yeah,
And we also
yeah,
have this kind of indirect way grounding the BioCipher knowledge graph in ontologies by having an instance-based knowledge graph on the basis of ontologies, which means not the entire ontology is in your graph, which plays a role if you, for example, want to deal with things like experimental factor ontology or disease ontology, where Uberon has like 500,000 concepts. So there are a bunch of ways of going about it and tying it to the constrained attention that LLMs still have, because you can't push an entire ontology into the model and then hope it kind of grounds correctly.
Yeah.
And then there's also the competing internal knowledge, because LLMs by definition bring their own world model, which is just derived from all the training data and the biases that the model has. And sometimes it conflicts with external information. So there's prompt engineering in whichever way you implement it, bringing an external ontology in, which is often also from the labels not as human readable as the everyday concepts that I think are better reflected in LLMs. You get conflicts because no human would talk in URIs. You don't say uberon.org/this large number. And since LLMs take a large part of their performance from us, from the way that we have communicated on the internet before, I suppose they have the same problems of dealing with these very non-intuitive identifiers. So there's a bit of tricking and engineering involved in making that work.
Melanie?
Yeah, I mean, as they are look and have human language as input data, which is unstructured, having structured data like you would have in these URIs, is probably not the easiest way to digest for the LLMs. I've seen other people who try to get LLMs to create a structured output like a JSON format or something, and it did something that looked like it, but actually couldn't consistently produce the same output because it just, yeah, eventually it's not what these models like and what they're good at. So structure, it's funny because we try to get structure into data and trying to use those models to structure our data, but actually they're trained on unstructured text from human language, which is, yeah, sort of it will hamper them in producing structured output, but also read in structured input. So they may not actually be happy with it. But if I may, I wanted to look at these other questions. So someone asks about cancer imaging and LLMs. So, I mean, to me, this is a computer vision problem. I don't know what the question here, whoever asked it, what they exactly want to do with the cancer imaging data, what it represents, if it's a description of the image in text format or if it's an image itself. Because if it's an image itself, I would just try with computer vision rather than actually LLMs at all. I don't know if anyone else has a response to that.
Yeah, it depends what exactly they're trying to do as well. So if you're trying to go from a chest X-ray to a radiology report, that kind of vaguely makes sense with a multimodal LLM. You can feed it an image and you can train it on image report pairs. That might work. But LLMs are designed for language primarily. And so I mean, obviously multimodal models work because GPT-4o is a thing and it can understand images and produce images. So you can get the transformer architecture to do this and the world model inside the model kind of can figure stuff out. But the depending on what you want to do, if you want to classify images, you throw that at CNN. You don't do that with an LLM. That's kind of horses for courses, really.
There's also this nuanced aspect of the pre-training that is giving the model the basic language facilities, and then what usually we call fine-tuning if we go into a specific domain and make it adhere to a specific behavior. And only in the cancer context, I mean, the question said cancer images, but we need some text to train. But I doubt that there is enough text, even in conjunction with radiology data, to get the basic language functionality that you have in something like GPT-3 even, because the text that you need is just enormous. And not any domain has enough text to convey a grammatical understanding on the level of typical nowadays LLMs. So you always need the pre-training, and the pre-training needs to be a huge non-domain corpus.
Yeah. Okay. Yeah. Thanks for bringing that one up, Melanie. So there's another one here. How reliable are LLM models for biological research? Where do you see them as a useful tool? What are ways that people use them that they should be particularly cautious about, and particularly cautious about trusting their results? Go on, Matthias.
I mean, they should be cautious about everything, literally everything. There's not a single LLM that can be 100% relied on. That said, we've did a long way since GPT 3.5. So no, in terms of are these LLMs useful, whether they're commercial ones or open LLMs such as Llama 3, they are extremely useful. For biological research, I mean, in my field, it's protein classification, and they're great. They're very useful to describe protein families, extract information from papers. That's pretty the easiest way to do that. Just give a lot of papers, several papers to an LLM, and instruct the LLM to extract this information. It's pretty good. And to streamline curation process, like checking data updates, they also start to be pretty good. Since it's a very narrow thing, what all curators do in InterPro, they're not there yet, but they're getting better and better. It's just a bit of an art to write the prompt.
I have a kind of generic answer to this, which is that my philosophy is you can use them to support tasks that you are very confident in evaluating the output that you get. But as soon as you enter the territory where you yourself cannot tell whether what you received from the model has any merit or is completely wrong or maybe half wrong or whatever, then it becomes very dangerous, and that is where you need to become very skeptical and maybe figure out a way to approach this in a more sustainable fashion, like breaking it down into sub-problems where you can determine whether the LLM made sense or not. Maybe even go to the length of defining a benchmark of the common things that you wouldn't expect it to do correctly. And then if it doesn't do well, you know that you can't trust it. And this keeps developing in the same vein that we said before, because the models change all the time. And they may be able to do something that they couldn't 6 months ago. So you again have this continuous monitoring problem where I didn't trust them to do things a year ago, and now I say, yeah, but there's not a huge chance that they get it wrong.
Yeah, I mean, if you have a problem that's well defined, and it doesn't really matter whether it's biological domain or anything else, these models are really, really good, and they serve you as an assistant. I always see them as an assistant to do something that is really sort of repetitive and boring, and they can just do that for me in a quick, streamlined way with reliable results. Anything that goes beyond that, I would be very cautious of the results, and also perhaps not even put too much emphasis onto developing them, because it may well be just too vague an approach, and the results are just too unreliable, and I simply wouldn't just, yeah, the investment that you need to then do in order to make them reliable is probably completely outweighing what they will return to you. So in that sense, and that applies to any domain, it's not just biological.
In a way, this parallels really the different roles in science as well. So we could as well talk about master students and PhD candidates at this moment, because you give them problems, you get them to do things that you don't have time to do. You hope that they come back to you with something reasonable. And then there's also like generations of them, and you train them, like you invest some time into them, and then you hope they become better over time. And I guess if you view it like that, and you always remain skeptical, and you discuss with them also whether what they did is reasonable or not, I think that is a better perspective on it than trying to replace your own facilities or whatever you want to remove from your table.
Yeah, I'd agree. Anything where so that all of these reasoning models that have come out recently have come about because you can they're basically based on maths and code benchmarks. So you can get it to generate the answer to a maths problem, and you can if they're right or not because it's maths. And similarly, code, you can compile it and see if it compiles or see if it runs and produces the expected output or whatever. So that's quite a well-defined, it's very clear if the LLM has got it right or not. Other things, so where you don't know the answer or the answer is in some way kind of fuzzy, like if there's some element of subjective judgment in it as well, that's the absolute killer for this sort of thing. It's just no hope. That's when it starts to get really tricky. Sebastian, yeah.
Yeah, since you mentioned that, there is a very interesting anecdote that came out recently. A new exploit that people have developed for this particular approach to code generation, which is called slob squatting in reference to the old typo squatting problem. So LLMs have been observed to always generate the same non-existing libraries to import when given a coding problem. And then people have gone and created these libraries on PyPI and injected them with malware. So that if you just take the code and compile it in the way that you just described, it will import something that does something to your system. And people have done it forever for very common typos that you make in, I don't know, import pandas or whatever. And now it's being applied to LLMs. So that is just some observation on the resilience of making that work.
Yeah, that's.
But I guess we can use LLMs to detect those malware libraries in PyPy because it's open.
Yeah, LLMs fighting LLMs. It can't end badly. If anybody spots one they want to pipe up on, please do.
Yes, I can have a quick comment on someone's asking about ever tried to test any LLM within patent documentation covering biomedical topics to establish annotations, properties, binding data, etc. So, I mean, this is a typical named entity recognition problem, and that doesn't matter whether it's patents or not. You have to have your test data, your training data. You have to know what you want to find, and then you don't even need an LLM for that. This is something you can do with a small transformer, pure transformer that will just do the named entity recognition. So it depends what you want, but it's any text data. So whether it's a book, as a paper, or a patent document, it is the same type of input data, and it works just as well, or not, depending on what you're looking for.
Yeah, I think any place where you've got text to analyze, you can apply this type of model, provided you can specify the problem clearly and develop that benchmark or test set or however you want to call it. So yeah, patents should work. Pretty much anywhere with text should work. So there's a question about LLMs having exhausted the amount of data they can be trained on. So how does that reflect on the usage of LLMs in bioinformatics research? Should we expect better from LLMs in terms of bioinformatics in the recent future, or did we reach a plateau? So I have a bit of an opinion on this, if I can start. So I think it's true that we are reaching a kind of diminishing returns for scaling at the pre-training stage. So models have gotten preposterously big. What was the Llama? What was it called? The biggest one was 2 trillion parameters that Meta just released, which is ridiculous. That's absolutely preposterously enormous. And it's trained on trillions and trillions of tokens. And it's not actually that good. Like it's pushing the boundaries, but we're not seeing the huge leaps that we saw from 3.5 to 4 to 4.5 and so on. So the pre-training side, things are slowing down a bit. But what we're seeing now with the emergence of reasoning models like O1, O3, DeepSeek R1 is inference time scaling. So this is where you have a decision where you burn your compute money. So you can burn all of your money pre-training the model for ages, or you can burn all the money generating stuff at inference time. And what I think is happening now is a bit of a shift in the scaling. So the overall amount of compute maybe is staying the same, but the way it's allocated is changing a bit. So people are using reasoning models which generate, and if you've ever seen some of these reasoning traces, they can be extremely long and kind of crazy to answer a simple question. But it's burning GPU time when you ask the question to try and make a better job of the answer. So I think what we're seeing is, yes, we've sort of plateaued at the pre-training. You can't just ask an LLM a question or to do something complicated and it will work. But if you then have the reasoning capability and you kind of burn a bit of money at inference time, you can still do better. And yeah, you can kind of see that in R1 and O1 and all of those things. So Melanie?
Yeah, a little bit of a different take on that. I mean, going back to text data, there have been recent studies that actually, in terms of biological knowledge that we publish, we've sort of plateaued for the last 10 to 20 years. So we're basically just repeating the same knowledge, writing papers about the same thing and focusing in about the same research areas. So what I would hope for is in order to overcome the lack of data, which is basically we just having reached the amount of text we have available and the amount of biological knowledge, if you just stick to the life sciences, is that now that we have models, we can actually start to create hypotheses and then start investigating that corner to then open up and write new things. That's where our creativity comes in. So perhaps it's just a plateau at the moment, and we need to open up the new areas in order to have then more text about novel things. So that would be my hope, because it's been demonstrated that also the language we're using, so for writing papers, has sort of been stalled. So we're just churning out over and over the same thing, reordering words in the sentence, but basically having the same content and the same number of or complexity of words we're using. So the language complexity has gone down, perhaps also because none of us are perfect native speakers in English, but vast majority of publications are in English. So the language itself has become simpler. So that is an underlying problem for the training data that then limits the models. And as I hope just that by now having this tool and asking more questions about things we didn't realize there were questions before, then retrospectively in a couple of years' time offers up more data.
Yeah, that's a hopeful take. Thank you. Sebastian?
Yes, this is definitely correct, and it applies to the use case where you want the model, one singular model, to solve a problem for you. And I think that is where the plateau is. And if I want to generalize what you said, Andrew, this kind of reflects the bitter lesson in the sense that we now have brute forced the way to a fairly stable performance plateau in LLMs. And the next stage will be to find mechanisms to disentangle the language proficiency, which is the main, for me, it's the main focus of an LLM. I want to have something that can be a human computer interface via natural language. And language they understand very well, even starting with GPT-4, I would say. So this communication device functionality is a given. And then what we change in terms of the bitter lesson is that we don't try to teach the same architecture, the same model that we're training in language to also understand bioinformatics, to also understand code, to also understand how an API works or something like this. But rather we go the way that is already being walked by facilitating them using other things. And the more general, in terms of the bitter lesson, we create this interface to other things, the more we can rely on them as the translation engine between what the user wants and what the backend, however that looks, is going to do. So model context protocol, for example, is a big step in this direction, because if you have a good model context protocol for a specific thing, then it doesn't matter what that thing is, the LLM will understand it. And as long as the LLM is capable enough to understand that there is a tool that it should use to achieve a certain goal, it doesn't matter what the tool is, at least very simplified. So I guess from these developments, we will see the next step in robustness and performance, rather than just putting more parameters, more training data, etc.
Yeah. Thanks, Sebastian. Matthias.
Yeah, there's two small comments. The person asked about the plateau of LLMs in general and also specific in terms of bioinformatics. So, in my opinion, and that is not necessarily research oriented, in terms of software development or scripting in bioinformatics, there is indeed a plateau, but that's just due to the limit of tokens, input tokens that are available compared to the number of tokens for general software development. Because yeah, LLMs have been trained on a lot of Java code, Python code, so they are able to create bootstrap application very, very fast. But in terms of bioinformatics workflow, for instance, using Nextflow or SnakeMake, the amount of available code to train on is rather limited. So this is why they still struggle to make something that makes sense and works at the first attempt. So this is why it is possible to create a fast API or Django application in only a couple of hours on Cloud. It's more difficult to make, a proper bioinformatics workflow using SnakeMake in the same amount of time. But in terms of research, that's pretty more what Melanie, was talking about, that we reached this plateau where we don't really publish more novel ideas in general. And a question for Melanie is that if you expect us to use our human creativity to find novel ideas, or to rely on LLM train on all those human knowledge to find the vision where we should go. Because at the moment, LLMs are not so creative and they're not very good at defining novel ideas.
It's not waiting for the LLM to suggest you something creative, but more like using it as a discussion partner for you, because if you provide, in particular, if you run it in a RAG setting or something, and you have large databases behind it, if you wanted to explore the content of such a database writing queries, you would be hard pressed to constantly write the queries, but it's much easier to use an LLM and asking in a natural language. And then basically just use that to probe and explore what's the coverage in, say, UniProt, and then see where are the gaps, where have been things, what's the what do they call it? Dark matter, dark protein? I can't remember what it was. So the things where we have no description, no experiments, no analysis. So what would be necessary in order to fill the gaps? And then you can get an LLM to help you design these experiments, and then you can go back and publish that, and then you therefore increase the coverage and the knowledge we have. But it always, makes it necessary that you are in the loop. You have to be the one to make the decision. What are you going to explore? What experiments are you going to try? But it will increase the reach rather than just following what isn't usually what people do. They're following some mainstream ideas because that's where funding goes, that's where money goes. But now you have actually a tool at your hand where you can sort of propose an approach reasonably quickly and and cost effective, because then LLM will assist you with that, and then you can explore the new areas that are currently completely blank.
And a quick comment on that, there are also companies that try to do what you described just now you shouldn't do. Future House being one of the more well-known ones. Basically autonomous science that has basically some robots in the loop talking with other robots and then pushing science forward. It's a very tech bro-y startup kind of thing to say. Predictions on whether it will work or not, your guess is as good as mine. I'm skeptical, but who knows?
So I was just going to bring up, there's a couple of companies that I've seen that have done AI scientists, which they just released that they got through peer review. at one of the major AI conferences. So double-blind peer review. The paper that was submitted was completely conceived by AI. The experiments were written and run by AI. The paper was written by AI, and the AI submitted it. So no humans were harmed in the making of this research, and it got through peer review at a very prestigious machine learning conference. So I think that sort of thing they can it's closed loop because everything is in silico. They can just do stuff. Life sciences is going to be a bit different. You need some interface to the real world, but I think these sorts of things are going to be coming, and it's going to be tricky to make sure that they're not doing potentially dangerous and scary things, I think.
I mean, that's why I say you, as the human, you have to be in the loop. You make the last resort decision. You can't just let the I know there have been people, even 10 years ago, they've been trying to build robotic wet labs where you have, and very primitive at the time, AI designing the experiments, monitoring the machines, and this kind of a thing. And yeah, they just go off scale eventually. So no, you make the last decision here.
Yeah. Okay, now I've just spotted one which I think is for Sebastian, so we'll ask you that very quickly before you have to leave. Thanks for the good discussion and opening for questions. I'd like to know what the biggest challenges that BioCypher is aiming to address in the future within its ecosystem development and what's in the outlook for the next stages.
Excellent question. So I think the single biggest challenge is the community, because we set out to be an open source repository of these workflows to democratize knowledge representation, as we put in the title of the paper. And the problem is not at all technical. The problem is making it discoverable, like applying the FAIR principles to the framework, which is a meta thing that sits on top of all the individual projects that you do. And then also facilitating every project that may be very different, seeing how different projects in science are. So you have a framework that supports any kind of project with knowledge management, ideally making parts of other projects reusable. So this flexibility, coupled with the fact that you need to maintain an active community, you need to make sure that people are aware of the thing, that they get in touch if they are aware, that you're able to help them once they've gotten in touch. It has worked a bunch of times, so I'm not dissatisfied, but it could be much better. And things like this webinar help a lot with this, with the dissemination approach and with telling people what it is about and what we want to achieve. So the next stages should be what I described earlier about the model context protocol. We would like to have a hub, which doesn't mean we want to collect everything in one place, but rather we want to make sure that everybody's able to find all the things that they may need for developing such an application, which is knowledge representation, LLM technologies, and the technical know-how to do it, and maybe talking to other people who have done similar things. So yeah, very social endeavor, I would say. That is a challenge.
Thanks. Okay, let's have a look what other questions we've got. We could maybe try and answer this one. So this is a little bit outside the standard application of LLMs, but have any of the panelists got experience doing zero-shot or few-shot learning scenarios for biological sequence classification? It's not something that I've tried. I would be surprised if that will work out of the box with a commercial or like a standard LLM that you could download. Matthias?
Yeah, no, I would be very, very surprised. I haven't tried it because it would even cross my mind to try that. There are much better tools to do this, especially like yeah, protein languages. LLMs are not. I don't think LLMs are a good fit for this. It's tempting, and I think it's because of the widespread of chatbots such as yeah, ChatGPT, Claude, etc. No, with Gemini, I think from Google. It's very tempting to just drop your data there and hope for some magic to happen, but the reality is just it's not like that. And I think that may have been the also kind of a sort of confusion between LLMs and chatbots that may have led the other person earlier to ask about cancer images. In the end, some people just look at a chatbot and just drag and drop the image, and they may think that yeah, that the chatbot's going to be able to analyze the images, but that's not how it works.
Yeah, Sebastian.
Yes, I have not tried it for similar reasons as Matthias said, but I've seen similar preprints at least, and they don't work. So this probabilistic next token prediction doesn't work in contexts that are not human language. And so they are not designed to give you sequences with meaning in any biological context. And epitope prediction, for example, is a particularly difficult one, even for dedicated algorithms. So there is no chance that LLMs will work from my humble perspective. Um, since I need to leave soon, I want to take the opportunity to thank you again for having me. It was a very nice discussion and seminar overall. I'm looking forward to the next one, hopefully,
Yeah,
and have fun with the rest of the discussion.
thank you very much, Sebastian. It's
Sorry
been
that I
great.
need to leave.
Thanks. Bye.
And one thing that actually LLM could work for some very specific protein sequence classification, and it's some short linear motifs because usually they are rely on regular expressions. So if you give a knowledge base of known short linear motifs with a regular expression, so that is the sequence of known amino acids that compose these motifs, then yes, you could have an LLMs detecting these motifs into the yeah in your sequences, but that's not better than just writing a simple regular expression yourself.
Melanie?
Yeah, just because we had not so much about the zero shot, but because we had the protein language models coming up. So actually, I wanted to pick on one of the questions we had pre-submitted. We also had a quick discussion before we started here. So that is about I'm unsure whether this fits the scope, but would it be possible to discuss protein language models? Do they share the same underlying principles as LLMs? And given the state of things, are they a viable method to pursue for the more sequence-centered questions? In terms of technology, so these models, there's no difference to protein language models to LLMs. So you have to consider that an amino acid or a base in your sequence, whether it's a nucleic acid or a protein sequence, basically represents a word. So the principle is rather than the next token prediction is not predicting a new word, but it's predicting either a nucleic acid or predicting an amino acid. And otherwise, the principles are the same. So you can develop these models just the same way as you would do with a human language, just using sequences as input. Yeah. I mean, if you have a sequence problems like that, yes, you can just develop it in the same way. And that's, yeah, I comment on that.
Yeah, so as Melanie says, the underlying architecture might be very similar. But yeah, an LLM, as we're just throwing that around, is a large language model which is trained on human text, natural language. So it will have seen some protein sequences around the place, but it's not intended to know anything about them, and it the amount of that data in its overall pre-training corpus is going to be so infinitesimally small that it won't have learned anything about protein at all. So yes, you're much better off if you want to do something sequence centric, going with one of the sequence pre-trained models. There are some slight variations in architecture actually. So one that I was looking at recently is the Evo suite of models, which have a slightly different attention mechanism, but it's just that everything else is the same. So it's tiny little tweaks in the architecture, but yeah, they're basically identical.
What if I want to reproduce something just with new data instead of predicting things? what do I have to tune in the parameters of the model? So by definition, if you give it new data, you either fine tune the model for new data, and then you have to go to fine tuning, or if depending on what you want to do, even retraining proper. But if you just or inference, so new data, so if you give it new data, you either do the inference on it or you do the fine tuning on it. So you can't reproduce results with new data. That's by definition. But yeah, so it depends on what you want to do. If you have a starting model and you've got similar data, but maybe let's say it's something from a different species and you want to optimize a model for a particular species or something for sequences. Honestly, I don't know what data you're thinking of, but then yes, you could fine tune a model for your particular data, and then you can only do the inference on that particular data. Or if you have the initial model yourself in-house, you could think of increasing the training data, so merging the original data and your own data and retrain from scratch. Then you have the capability of both data backgrounds in your model. But yeah, so these are options you can try. It depends on what you want to do, and it depends on what model you're after, what your data is.
Okay, thanks, Melanie. What about standardized ways to normalize transcriptomic data when we are establishing prediction models to predict survival by using that data from a public database? What are the criterion to evaluate the performance of different machine learning models when developing prediction models? So this isn't really LLM specific.
Yeah, no, this seems more like a normal post RNA-seq pipeline, and yeah, normal normalization technique at TPM, RPKM, which is basically a question that could be easily answered by ChatGPT or something like that. But yeah, there is no LLM involved in this
Yeah.
thing.
Yeah, I don't think there's a role for LLMs and stuff like that either, because they'll just suggest things which are it's the same thing that Sebastian was saying, where if the answer is something that you can't check or that you're not sure whether it that you've got no kind of intuition whether or not it's right, then you really shouldn't be using an LLM. And so getting an LLM to suggest things for a survival model sounds extremely risky to me.
Well, there's a high chance it's just going to spit out whatever you may have seen on BioStar or other bioinformatics community forums. But the same way that if you just copy and paste that question into Google, probably the AI overview from Google is going to give you something very similar. But yeah, asking that on proper community bioinformatics community channel would be a better thing to do.
I'm happy to respond to Sumit's question. What is the future of bioinformatics as a career? And I think it will stay. It will just change. It will require you to, um, you know, work with those models as a partner, as a co-investigator, perhaps, someone you kind of like have as an assistant on the side, because just relying on your human performance will not be matching the data and the information we have. And so I think in that regard, you have to be comfortable with those models. You have to understand them to understand what they produce, to be able to judge what the quality is that they're giving you back. But your job as a bioinformatician will stay. It will just slightly change in scope and in topic. But there is a future, and I would go for it.
It's also different again based on what are you doing in bioinformatics. If you're like a faculty members from a researcher, a principal investigator, yes, your job is pretty not at risk because you have a PhD, because you have a lab of students and postdocs, and yes, you're going to use LLMs as assistant. If you are a code monkey writing scripts and pipelines. Yes, you need to adopt these ones and fast. Otherwise, someone else is just going who already adopted those ones is just going to come and take your job.
Yeah, it's the same thing of your job won't be taken by an AI, it will be taken by someone using an AI, right? The people who adopt the more productive tools will kind of be more likely to stay in the job. And it's the same throughout everything, I think. Okay. There's one question about using graph ontologies to help build tools and reduce complexity. So I'm not completely sure I understand this, but there is. So Sebastian was saying about how if you just throw thousands of tools at an LLM, it gets confused and doesn't know what to do. There are efforts to do retrieval augmented tool selection, basically. So rather than here's a huge pile of tools, try and figure out what to do with them, you would query a RAG database that has a description of all the tools, and then it will pull back the most likely, like top 5 tools that might do what you want, which is an interesting direction. I think people are kind of exploring a little bit. And then once you're talking about RAG, you can talk about graph RAG and kind of more fancied versions of it as well. So yeah, I think it can help reduce the complexity of selecting a tool. I don't know if it helps with actually making the tools.
Yeah, I mean, I didn't quite understand the question either, but if you're just thinking of using ontologies as a way of um, reducing complexity for your LLM and giving it some guidance and some framework, then yes, that's what we said that before. You can use ontologies, turn them into a knowledge graph, or directly use ontologies depending on what your setup is, and then use them for RAG and give your LLM some guidance. But it depends on what you want to do.
Okay. We got one new one. There are a lot of web-based AI tools. How do you trust them? You don't. So I'm a big proponent of local LLMs. I don't use APIs for anything anymore, because you don't have full control over the model. So if you're talking to ChatGPT behind the scenes, it's generating a probability distribution of tokens, and then there is some intermediate process which selects the token that it actually gives back to you. You don't have any control over that if you're just using some random web tool, which means you don't have any control over the temperature, any of the sampling parameters. You can't do constrained generation that we talked about. You can't force it to do anything. You're completely at the whim of whoever programmed it, having chosen sensible defaults that will work for your case, which is incredibly unlikely to be true. So I would take pretty much everything from a web AI tool with a massive pinch of salt. Melanie?
Yeah, I mean, my background as structural biologist has been, that I want to look at a structure and find out what are the specific features and what are the important bits. And there have been lots of people who've been, using PDB as the underlying database, and then writing an LLM application, chatbot kind of a thing on top of that, and trying to sell it to you as a way of visualizing your structure and talking about it. And the simplest thing you can do is point me to the active site residues in my protein and give it a PDB entry, and it will fail massively. And that's done so over and over again because it doesn't do the analysis of the structure. It does not understand what a 3D model of a protein is. And I've just had that so many times that when I get the next email in my inbox, we've got this great model about the PDB. Can you please have a look at it? And, do you want to host us on your webpage? Or just laugh, just delete the email because it never works. Because, we have been trying to do that in-house as well. And it's such hard work to do that, yeah, that it's not an easy way.
Is
So,
it
yeah, go on, Matthew.
so? Is it also because people train or fine-tune LLMs, they expect them to do that work when a simple machine learning or deep learning tool would be much better? I mean, I know that LLMs are deep learning, but the idea is that yeah, if you want to analyze PDB data, don't build a LLM, build a deep learning tool. It's much better.
Yeah, there's inductive biases in the choice of algorithm, right? Like you can choose a better algorithm than autoregressive text complete to do these things. So why would you? You've got a hammer and you think everything's a nail if you've got one of these LLMs. Okay, well, thank you both. I think that's all the questions. Call the end of the webinar and the end of the series a little bit early. So I just want to thank once again all of the panelists. It's been absolutely brilliant. Thank you all for being so game to do a talk and then subject yourselves to being grilled for an hour and a half. Thank you very much. This would not be easy without other people to help. I hope you've found this useful in the audience.